Hi, everyone. My name's Katie Primm, and I'm a new hire at PSI. I started roughly in the spring. And today, my talk is studying streaks and the potential for water on Mars. So, my overall scientific goal from my research, from my PhD up until now is there life on Mars? But in order to understand if there's life on Mars, we have to go back to what we know about life. So, life as we know it needs liquid water. Fortunately, on Mars, we know that there's a solid and gas phase of water present. And you can see on the rightmost image, the Hubble telescope shows images of the North Polar Cap. And you can see that it's made of CO2 and water ice, and it disappears and it turns to just a water ice cap. And from the temperatures of that polar cap, we know that it is water ice and not just CO2. And then the image right below it, that's half cropped out, is a picture that the Viking lander took of the surface. And you can see it's kind of white casted. And from this image, we know that there's frost on the surface. And knowing that there's frost on the surface, we know that there are two phases present of water. So there's a gas phase and a solid phase. And then 2008, in the Phoenix lander, discovered a perchlorate ion. And that was pretty exciting because a perchlorate ion mixed with some type of metal, so like a salt, can have the potential for liquid water to exist because it has hygroscopic properties. So it's very water loving. It takes low relative humidity to turn into a liquid solution. So an image below the Viking lander image is the 2 scoops that the Phoenix lander took. And that's the 2 scoops that it found the perchlorate ions in. And then to the right of that image is a radar image taken from Marsus. And it's more recent, in like 2018, they found that there was a subglacial lake in the SPLD, or the South Polar Layer deposits. And it said that this subglacial lake is probably salty because of how cold it is down there. So evidence for some type of salt and maybe liquid is present on the surface of Mars. And then lastly, a lot of traction are streaks on the surface of Mars. So recurring slope lineae, or RSL, and slopestreaks look like liquid flows, but we aren't sure. So we want to study them to see if they are liquid flows. Maybe they're everywhere. If not, what are they actually made out of? So for my PhD, I looked at Raman microscopy of different phase transitions of certain perchlorate salts. So here is the magnesium perchlorate phase transitions on the left. So it's just intensity versus wave number of this perchlorate going through different phase transitions. So on the left, it's just the OH stretches. So it's going from the hexahydrate, so that black tiny peak, and then the red one is its deliquescence. So now it's in the aqueous phase, and then the ice is the green phase, or sorry, the green color. And then it goes back down in phase all the way down back to the hexahydrate. So you can see on the right, it's the perchlorate, just showing that we are looking at magnesium perchlorate, but nothing really changes in the perchlorate peak. So with my laboratory studies, I can see what phase we are looking at for magnesium perchlorate in the conditions of Mars. And then on the right image, you can see a phase diagram. So it's the relative humidity versus temperature of magnesium perchlorate. And the gray dots that are kind of faded, those are the laboratory points that I've found in the laboratory. And then all the dotted lines are modeled phase transitions. So the deliquescence line is in the middle, where it turns into the aqueous phase, and then the ice line on the left. And then efflorescence is just the recrystallization from the liquid phase. So I wanted to put my laboratory experiments up next to actual Phoenix rover data, or Phoenix lander data, to see how's that look. Is it going to have liquid? Is it going to just be ice? Is what's happening? So if you look at the black circles, you can see that it's the TCP data, just the in air, and it doesn't really reach any of the liquid phase for magnesium perchlorate. But we had a modeler modeled the subsurface, so 1 centimeter below the surface, and that's in red. And you can see that it passes through the liquid phase. So potentially on the landing area that Phoenix landed on, there might be liquid water in terms of magnesium perchlorate brines. So the next time for my PhD, I wanted to look at changing the conditions. So in my previous one, I can change the relative humidity and the temperature, but not the pressure. And so for my PhD or for my postdoc, I wanted to make a new Mars chamber that can look at pressure, because that could be an important part of looking at liquid water on Mars. So here I built a pretty complicated Mars chamber, but all in all, I'm saying that I can change the pressure, relative humidity, and temperature while looking at the electrical properties of samples. So that can also tell you if there's liquid phase or an ice phase present. So on the top right, I'm just showing you what's inside the Mars chamber. So we have our sample holder sitting on a liquid nitrogen cold plate, and then some fans and temperature sensors. So just noting that our sample for this chamber is bigger than the sample in my Raman microscopy, because Raman is spot size around 1 micron. And this one is 20 millimeters by like 5 millimeters in height. So using this Mars chamber, I then took some data with magnesium perchlorate, again, mixed with fine sand in different percentages. So I just want to focus on the 2.5% magnesium perchlorate plus fine sand. So that's in the gray. And with these, I looked at the time elapsed in days versus relative humidity, temperature, volume of brine, and dielectric permittivity. So dielectric permittivity can tell you if liquid is present, and then you can calculate the volume of brine from that. So if you can see, in the beginning, I was at 50% RH. The dielectric permittivity was quite high, and then the volume of brine is high. And then I dried it out and then rehumidified it up to 50%. So that's at around a day, a little after a day. And you can see that the volume of brine increases as the dielectric permittivity increases. But even though deliquescence or brine starts quickly, it takes about a day to complete to full saturation. So this is still preliminary. This is so far what I've gotten, and we're still working. So I'm working with David Stillman at SWERI to do more of this, to see how long does it take? Is it dependent on the salt? How much salt? All the stuff that we're trying to look at. So next, I want to talk about flows on Mars. So usually when people think about flows on Mars, they think about RSL. And RSL are recurring slope lineae on the bottom left. So they recur seasonally every year. And then on the right are Slopestreaks, which are about 10 times bigger than RSL. And they're not seasonally. They sometimes fade. Most of the time, they don't see them disappear completely, and they form, but we haven't really seen them fade in a seasonal fashion. So they're pretty interesting because we don't know what's going on. So I started looking at Slopestreaks, and luckily I won my first PI grant with the Mars Data Analysis Program, and I'm studying the formation and fading mechanisms of these Slopestreaks. So on the bottom left here are just an image of the Slopestreaks in the region, one of the regions I'm going to be looking at in my grant. So what started me looking at Slopestreaks more in depth, because a lot of people haven't focused on them as much, thinking that they're dry flows. But on the image on the left I show here I've circled a couple of Slopestreaks in red. And then on the right side, something cut out, but okay, let's move on. So here I'm doing a comprehensive study of Slopestreaks. So here I'm looking at Slopestreaks, the angled or the sloped area right next to it, number 2, and then the flat area in number 3. And then I take ratios of those to actually figure out if the Slopestreak is fading. And so the C graph on the right is showing the ratios, and then the green line is the difference. So you can see if it's fading, it's going to be a downward slope. If it's getting darker, it's going to be a positive slope. So here we know that this is fading without any visual bias, because if you look at it just with your eyes, it's hard to tell. And then next, my co-eye Rachel Hoover is going to make a DTM of the slope angles. And then my co-eye Hannah Kaplan is looking at CRISM images to see if there's anything interesting spectrally. So we're going to try and get a comprehensive study of slope angle, fading, and forming ratios, and then CRISM spectral images. So lastly, since I'm a new hire at PSI, I'm looking for collaborations. So again, to reiterate, my scientific goal is astrobiology, looking for life on Mars and different planets. Mainly I've done Mars, but I've dipped my toe into Europa a little bit. And I'm really interested in Ceres and Enceladus. And what I have to offer, if you've forgotten, I am well versed in IR and Raman spectroscopy, vacuum chamber systems, surface atmospheric interactions, and MRO image processing. And some groups that I want to work with that I want to do more with is, of course, working on missions because I haven't been on one, and more data processing for satellite and rover data. And thanks.